Good morning, grade 8 students. This is Miss Moni. Today is March 19, 2020. We are going to continue chapter 3 circulation, lesson 3 blood and lymph. So please open your books to page 91. The objectives of this lesson: we are going to identify what determines the type of blood. We are also going to explain how safe blood transfusions are made. To recap, blood vessels transport the liquid that is vital to our survival. Blood is not just the plain red liquid that passes through our blood vessels. It is actually a complex tissue that has several parts. Blood is made up of plasma, red blood cells, white blood cells, and platelets. About 45% of the blood is cells, and the rest is plasma. Blood types. We have several questions to ask when we talk about blood types. How many blood types do we have? Can we transfer blood easily and safely from one person to the other? To answer these questions, we should first study about blood transfusion. What is blood transfusion? Blood transfusion is the transfer of blood from one person to the other. Sometimes the two blood samples blend smoothly, and in other cases the red blood cells clump together. This clumping may clog the capillaries and may lead to death. Why this happens? To begin with, we should know that we have 4 blood types, some of which are compatible with each other and some are not. The 4 blood types are blood type A, blood type B, blood type AB, and blood type O. Marker molecules. The blood type is determined by a type of protein called the marker molecule found on the red blood cells. Each type of blood has a specific marker molecule that determines the blood type. Blood type A has a marker molecule A on the red blood cells. Blood type B has marker molecule B on the red blood cells. Blood type AB has both marker molecules A and B on the red blood cells, and blood type O has no marker molecules on the red blood cells. The figure below shows each blood type and the marker molecule that it has on its red blood cells. To answer the question why blood clumps sometimes while doing transfusion, because your plasma contains also clumping protein to make safe blood transfusions. These clumping proteins recognize foreign red blood cells and marker molecules on the red blood cell and make those cells clump together. This figure shows that each type of blood has a clumping protein. For example, blood type A has a clumping protein anti-B, so it cannot receive any B blood type. Blood type B has an anti-A clumping protein, so it can never receive a blood type A. Blood type AB has no clumping protein, so it can receive any blood type. Blood type O has a clumping protein of anti-A and anti-B, so hence it cannot receive any blood type A or B. How safe transfusions are made. The marker molecules on your red blood cells determine your blood type and the type of the blood you can safely receive. For example, blood type A can safely receive from an A blood type and O. Blood type B can receive from B and O. Blood type AB can receive from A, B, AB, and O, and it is called a universal receiver. Blood type O can only receive from O, but it can give all the blood types, so it is called a universal donor. Safe transfusions. The marker molecules on your red blood cells determine your blood type and the type of blood you can safely receive. There is a condition for blood transfusion to succeed. The marker molecule of the donor should be different from the clumping protein of the recipient. For blood transfusions to be safe, there is a condition to succeed. The marker molecule of the donor should be different from the clumping protein of the recipient. For example, if a donor of blood type A having a marker molecule A on its red blood cells gives a recipient of blood type B, also blood type B has clumping protein anti-A, the blood will not blend smoothly and hence it will clump and the transfusion will fail. The Rh factor. There is another protein found on the red blood cells is called the Rh factor. The presence of the Rh is determined by a marker molecule on the red blood cell. If your blood type is Rh positive, hence your red blood cells have the Rh protein. If your blood type is Rh negative, hence your red blood cells lack the Rh protein. Rh negative can only receive Rh negative blood, or else a clumping Rh protein would develop in the plasma, and this is dangerous. However, Rh positive blood can receive blood either from positive or a negative Rh blood. As a conclusion, blood type is determined by marker molecules found on the red blood cells, and safe transfusions are determined by clumping proteins also found on the red blood cells. This table shows each blood type, the marker molecule it contains on the red blood cells, the clumping protein it contains, and the blood that it can safely receive. As an assignment, please study pages 94 to 96 from the book and check the slides below to highlight the required information on your books. Thank you.